Hi, my name is Stephanie Paris, and today I'm going to talk about the process of mitosis and meiosis. Mitosis. It is a process of cell duplication in which one cell divides into two genetically identical daughter cells. The stages of mitosis: the prophase, metaphase, anaphase, telophase, and the cytokinesis. Prophase. This stage prepares the nucleus for the separation of two complete sets of chromosomes into new nuclei. It is important for subsequent mitotic events such as spindle assembly and positioning as well as chromosome capture. Metaphase. The chromosomes are arranged at the equatorial plate or middle region of the cell. The alignment of the chromosomes with sister chromatids on each side of the metaphase plate ensures the two new cells will be identical. Anaphase. The stage of mitosis after the process of metaphase, when replicated chromosomes are split and the newly copied chromosomes are moved to opposite poles of the cell. It is a very important stage of the cell division because it ensures that duplicated chromosomes or sister chromatids separate into two equal sets. Telophase. The telophase begins as soon as the daughter chromosome reaches the opposite end of the pole. And the cytokinesis. Cytokinesis is the physical process of cell division, which divides the cytoplasm of a parental cell into two daughter cells. Cytokinesis performs an essential process to separate the cell in half and ensure that one nucleus ends up in each daughter. Cytokinesis, animal mitosis versus plant mitosis. In animal mitosis, centriole and aster are exist, while plant mitosis, no visible centriole or aster. And in animal mitosis, the daughter cells separated by cleavage furrow, while in the plant mitosis, daughter cells separated by cell plate. Okay, let's proceed to meiosis. It is a type of cell division that reduces the number of chromosomes in all parent cells by half and produces 4 gamete cells. Meiosis is important because it ensures that all organisms produced via sexual reproduction contain the correct number of chromosomes. Meiosis phases. Meiosis 1 begins with one diploid parent cell and ends with two haploid daughter cells, having the number of chromosomes into each cell. Prophase 1. Prophase 1 is essentially the crossing over and recombination of genetic material between non-sister chromatids because this results in the genetically identical haploid daughter chromatid cells. The pairing of chromosomes occurs in a process called synthesis, which is not random. Rather, chromosomes that carry similar sets of genes come together and lie side by side. Metaphase 1. In metaphase 1, the homologous pairs of chromosomes align on either side on the equatorial plate. The nuclear membrane disappears and the spindle appears. Anaphase 1. In anaphase 1, the homologous are pulled apart and moved apart to opposite ends of the cell. This is stage characterized by the movement of chromosomes, both poles of the meiotic cell, via a microtubule network known as spindle apparatus. And telophase 1. Telophase 1 is that phase when the chromosomes have finished moving to opposite ends of the cell now undergoes a process called cytokinesis that divides the cytoplasm of the original cell into two daughter cells. Meiosis II. During meiosis II, the sister chromatids within the two daughter cells separate, forming four new haploid gametes. Prophase II. During prophase II, chromosomes condense and the nuclear envelope breaks down, if needed. The centrosomes move apart, the spindle forms between them, and the spindle microtubules begin to capture chromosomes. And metaphase. In metaphase II, the chromosomes line up individually along the metaphase plate. In short, spindle fiber appear and the chromosomes align themselves at the equatorial plate. And anaphase II. The centromeres separate and the sister chromatids, now individual chromosomes, move toward the opposite poles of the cell. In anaphase II, chromosomes divide at the centrosome, and the resulting chromosomes, each with one chromatid, move toward opposite poles of the cell. Telophase II. In telophase II, nuclear membranes forms around each set of chromosomes, and the chromosome decondense. Cytokinesis splits the chromosome sets into new cells. This will be followed by cytokinesis, wherein each of the two cells produced from meiosis I will give rise to daughter cells, resulting in a total of four genetically similar haploid cells. And that would be all. Thank you and God bless.